Okay, so this is the information that you need for Wednesday. It is the slides about the cell membrane. Technically, it's part 3 of this unit, but it's the first set of slides just on the cell membrane. You already looked at organelles. The cell membrane is very dynamic. We know it's made up of phospholipids, and the membrane is what we call semi or selectively permeable, meaning that certain things can go across and certain things cannot, and they need some sort of transport. This is an example of the phospholipid membrane. When we see extracellular fluid, that's fluid outside of the cell. Cytoplasm is inside of the cell. This is what I try to draw when I try to show you guys what a phospholipid looks like. This is just a much better image of that. Phospholipids look like the image ahead, but what's important is that it's made up of a phosphate head and hydrocarbon tails. They are made up of these phospholipids, so phosphate lipid molecules. The head is charged, so it's polar, and the tails are nonpolar. We also would determine that this has hydrophilic and hydrophobic parts. The charged phosphate head is hydrophilic or water-loving, and the hydrophobic parts are water-fearing. The reason that they're arranged in such a way is so that both of the hydrophobic parts are pointed towards each other, and the heads are pointed either outside of the cell or into the cell. This means that they can both the outside portion and the inside portion can interact with water, whereas that inside layer where like the legs are doesn't interact with water or polar liquids. They have the ability to move, so these phospholipids are kind of more like fluid. They move sort of laterally, so they have this like wave-like flowing motion. They rarely will like turn and face the opposite direction, but they can move a little bit. Membranes are more fluid when they contain more unsaturated fatty acids within their phospholipids. So more unsaturated fatty acids results in increased distance. So if you look on the left, we see all these spaces, and that's actually where fat would be. And so it makes the membrane more bendable and foldable and fluid. Whereas when they're more tightly packed, they'll move in a very different way. Sometimes we see that cholesterol, a type of lipid, is going to be found in between my phospholipids, and that will change the way that my membrane works. It affects what we call the fluidity of the membrane, or how it moves and how it allows things through. Proteins are going to be found embedded in the membrane. So we are going based on this thing called the fluid mosaic model, which is basically that we have this membrane, and if you've ever seen like a mosaic or like a stained glass window, it has all these different like things in it that have different important parts. A membrane protein, and one type would be a transport protein, basically transport molecules from inside to outside or outside to inside, and they're usually ones that can't pass normally. A specific type are called aquaporins that allow water to move across the membrane because, as we know, part of the membrane is nonpolar, so water can't move all the way through on its own. Other types of membrane proteins, so we've got our transport proteins, enzyme proteins. These allow some sort of reaction to speed up. Receptor proteins, these are going to be for hormones and neurotransmitters to send a message inside of the cell. The fourth type of protein are cell-to-cell recognition. They basically allow another cell to determine what they're bumping up against. And then intracellular joining proteins physically anchor a cell somewhere. Again, when we talk about the fluid mosaic model, this is basically looking at how we have our 2 layers of phospholipids, and then we've got all these different types of proteins embedded inside of it. They basically looked at a cross-section of the cytoplasm layer, so the layer closest to the inside of the cell, and they saw that it was pocketed with all these different types of proteins that were used for the things that we talked about before. Plasma membrane synthesis. So we have this phospholipid layer, but sometimes it's damaged. Sometimes we need to add more. And so the size and the amount of membrane can be increased or decreased when we create vesicles. If we remember back to the Golgi apparatus and the rough and smooth endoplasmic reticulum, they have things that will join and then basically spread out to become membrane, and it can increase the surface area of the plasma membrane. This is what the plasma membrane looks like up close. And that is it. There's another set of slides. It'll also be like 4 or 5 minutes.